Hello and welcome to my channel. In this video we will be looking at atypical gender development, gender dysphoria, biological and social explanations for gender dysphoria, which you need to know for AQA A-level psychology in the subtopic of gender. I hope you enjoy this video and find it helpful. Let's get started. Gender dysphoria, gender identity disorder, is a psychiatric condition where there is an inconsistency, conflict, mismatch or incongruence between one's assigned gender, on the basis of external sexual characteristics, and the expressed gender or psychological perception of self as male or female. A person whose biological characteristics are typically male will nevertheless feel female, or a person with typically female biological characteristics will nevertheless feel male. A person may experience strong and persistent feelings of identification with the opposite sex, resulting in depression, anxiety, and stress, due to a person feeling discomfort with their own assigned sex. Because of this, gender dysphoria is recognized as a psychological disorder in DSM-5. To resolve this discomfort, people may identify as transgender or undergo gender transitioning and reassignment surgery. This changes the person's sexual identity through changing their reproductive organs and hormone levels, which consequently changes their appearance. This allows them to more comfortably open dress and use mannerisms associated with the opposite gender. Biological explanations of gender dysphoria. There are 4 biological explanations of gender dysphoria: brain sex theory, genetics, phantom limb and crossing wiring theory, and hormones. Brain sex theory. Brain sex theory that gender dysphoria is a result of brain structures that are incompatible with a person's biological sex. Some areas of the brain are dimorphic, meaning they take a different form in males and females. This theory assumes male and female brains are different, and those suffering from gender dysphoria have brains that do not match their biological sex. One area of the brain that has been studied is the bed nucleus of the stria terminalis, BSTC, which is located in the thalamus. The BSTC is on average twice as large in heterosexual men as in heterosexual women and contains twice the number of neurons. Brain sex theory proposes that for people with gender identity disorder, the size of the BSTC correlates with preferred sex rather than biological sex. A strength of the brain sex theory as a biological explanation of gender dysphoria is that it is supported by research. Ju et al., 1995, used postmortems to compare the brains of 6 male-to-female transgender people with cisgender male and female controls, and Kruijver et al., 2000, compared the number of neurons in the BSTC regions of transgender people with cisgender people. They both found that the number of neurons in the BSTC of male-to-female transsexuals was similar to that of the females. In contrast, the number of neutrons in a female-to-male transsexual was found to be within the male range. This is a strength because it suggests that people with gender dysphoria have a BSTC which is the size of the sex they identify with, not the size of their biological sex. A strength of brain sex theory as a biological explanation of gender dysphoria is that there is research to suggest that there may be other brain differences associated with gender dysphoria. Rametti et al., 2011, studied another sexually dimorphic aspect of the brain, that of white matter, the deepest tissue of the brain. There are regional differences in the proportion of white matter in male and female brains. The brains of both male and female transgender individuals were analyzed before they began hormone treatment as part of gender reassignment. They found that the amount and distribution of white matter corresponded more closely to the gender the individuals identified themselves as being rather than their biological sex. This is a strength because it suggests that the brain structure of transgender individuals may align more with their experienced gender than their biological sex, supporting the idea of a neurological basis for gender identity. A limitation of the brain sex theory as a biological explanation of gender dysphoria is that it has been heavily criticized by other psychologists. Chung et al., 2002, suggested that the differences in BSTC volume between men and women did not occur until adulthood. Most people suffering from gender dysphoria report that their feelings began in early childhood. Lawrence, 2003. This is a limitation because it suggests that the differences found in BSTC are not the cause of gender dysphoria but an effect. A limitation of the brain sex theory as a biological explanation of gender dysphoria is that there is research to contradict its theories. Huselshoff Paul et al., 2006, used brain scans to measure the brains of transgender individuals before and after cross-sex hormone therapy. They found that transgender hormone therapy affected and changed the size of the BSTC. This is a limitation because it suggests that the changes in brain structures may be an effect of hormone therapy rather than a cause of gender dysphoria. Transsexual gene. The genetic explanation suggests that there may be a specific gene, or combination of gene variants that cause or make a person more vulnerable to developing gender dysphoria. A strength of the genetic explanation as a biological explanation of gender dysphoria is that it is supported by gene association research. Hare et al., 2009, examined the DNA of 112 male to female transsexual volunteers and 258 controls. They found that they were more likely to have longer versions of the androgen receptor gene compared to heterosexual controls. This genetic abnormality leads to reduced action of the male sex hormone testosterone, a critical hormone for masculinizing the brain during prenatal development. They concluded that the reduced testosterone activity associated with this gene variant can result in the undermasculinization of the brain, aligning brain structure and function more closely with female patterns. This is a strength because it suggests that there is a link between genetics and transsexualism. A strength of the genetic explanation as a biological explanation of gender dysphoria is that it is supported by twin study research. Coolidge et al., 2002, assessed 157 pairs of twins, 96 MZ and 61 DZ, for evidence of gender dysphoria. They found a 2.3% occurrence of GID. Of these variance cases, 62% could be accounted for by genetic factors. Halens et al., 2012, studied 23 identical, MZ, and 21 non-identical, DZ, twin pairs, where one twin had been diagnosed with GID. They found a 39% concordance rates for MZ twins compared to 0% for DZ twins. Diamond, 2013, combined a survey of transgender twins with a review of published studies on transgender twins. Concordance rates for gender dysphoria among identical male and identical female twin pairs were 33.3% and 22.8% respectively. In contrast, the concordance rate among non-identical twins, male or female, was only 2.6%. This is a strength because these pieces of research suggest that there is a heritable and genetic component to gender dysphoria. A strength of twin study research support for gender dysphoria is that their findings and conclusions suggest that gender dysphoria has some genetic basis. Twin studies have been found to show higher concordance rates for gender dysphoria compared to the general population. Research has shown that identical twins, who share 100% of their genetic material, have higher concordance rates for gender dysphoria compared to fraternal twins, who share about 50%. This is a strength because it suggests that there is a hereditary component to gender dysphoria, indicating that genetic factors may contribute to an individual's risk of developing the condition. A limitation of twin study research support for gender dysphoria is that the findings and conclusions may be due to environmental factors. The higher concordance rates found for identical twins may be due to nurture. Identical twins often grow up in the same household and share similar environments, which can make it difficult to separate genetic factors from environmental ones. This shared environment can lead to an overestimation of the heritability of a trait or disorder. This is a limitation because it suggests that the apparent genetic influence indicated by twin studies might be inflated due to shared environmental factors, making it difficult to accurately determine the true extent of genetic contribution to gender dysphoria. A limitation of twin study research support for gender dysphoria is that it cannot be concluded from these studies that genes alone determine who develops gender dysphoria. The concordance rates in twin studies are not 100% and therefore not all twins share gender dysphoria tendencies despite having the exact same genetics. This is a limitation because it suggests that genes only do not create a vulnerability and are not a direct cause, as other factors must trigger the condition. A limitation of twin study research support for gender dysphoria is that it lacks population validity. Twins are often selected from specific regions or backgrounds for a specific purpose, composing a sample of twins from similar socioeconomic statuses, cultures, or geographical areas. This does not account for factors, such as environmental influences and different cultural contexts that could affect the development of gender dysphoria. On top of this, being a twin is somewhat rare, with 1 in 42 children being born a twin. This makes the sample pool small, as the probability of finding a pair of twins from every single type of socioeconomic status, culture, and geographical location is rare. This is a limitation because it suggests that twin study samples do not represent the wider population, which can limit the generalizability of the findings from these studies. Phantom limb and crossing wiring theory. Ramachandran and McGeoch, 2007, proposed the theory of phantom limb and cross-wiring as an explanation of gender dysphoria and suggested that the image of particular sex organs is innately hardwired into the brain in a manner that is opposite to the person's biological sex. In the case of a male with gender dysphoria, they feel they should not have a penis, whereas in the case of a female with gender dysphoria, they feel they should have penis. It is estimated that 2/3 of female-to-male transsexuals report the sensation of a phantom penis or phantom erections from childhood onwards. A strength of the theory of phantom limb and cross-wiring as a biological explanation of gender dysphoria is that Ramachandran and McGeoch, 2007, was able to conduct research to support his theory. Ramachandran and McGeoch, 2007, used a sample of patients who had had surgery to remove sex organs, e.g. for cancer, and compared and examined the patients who had gender dysphoria and the patients who did not have the condition. They found that out of the patients who had had a penile amputation, approximately 60% who did not have the condition experienced the feeling of a phantom penis compared to 30% of those with gender dysphoria. This is a strength because it suggests that the individuals with gender dysphoria had no pre-existing wiring to a penis representation in the brain to begin with. Hormonal explanations of gender dysphoria. The hormonal explanation of gender dysphoria suggests that prenatal, in the womb, hormone levels can affect a person's likelihood of developing gender dysphoria. In females, an overexposure to androgens during prenatal development can lead to masculinization. In males, an underexposure to androgens during prenatal development can lead to feminization. This can affect brain development, structure, and function. Some possible outcomes include altered sexual orientation, increased or decreased aggression, or differences in cognitive abilities. This exposure to androgen hormone levels can be affected by the mother's own endocrine system, maternal stress, or illness. A strength of the hormonal explanation as a biological explanation of gender dysphasia is that the explanation is supported by research. Congenital adrenal hyperplasia, CAH, causes increased testosterone exposure in the womb. Erickson-Schroth, 2013, reviewed statistics on individuals with congenital adrenal hyperplasia, raised male or female. She found that of biological females with CAH, although the majority, 94.8%, raised female later developed a gender identity as girls or women and did not feel gender dysphoric. 5.2% developed gender dysphoria and were uncomfortable in the sex that they were born, which is higher than the prevalence of female-to-male transsexuals in the general population of chromosomal females. This is a strength because it suggests that prenatal testosterone exposure increases the incidence of gender dysphoria in biological females. Biological evaluation. A strength of biological explanations of gender dysphoria is that they can be socially beneficial. If a biological cause is found to affect gender dysphoria, then this could help members of society be more accepting towards the conditions, as they may accept that it is not the individual's fault that they are suffering from the condition and feel the way they feel in their own body. This can be a relief for individuals who feel out of place and abnormal because they were born in the wrong body. This could cause gender dysphoria to be classified as a medical condition, which requires treatment and removes the responsibility from the individual and gives them more of an opportunity to get help and support. This is a strength because it suggests that identifying a biological basis for gender dysphoria could significantly reduce societal stigma and increase acceptance towards individuals experiencing this condition. A limitation of biological explanations of gender dysphoria is that they can be social sensitivity. If viewed as purely biological, gender dysphoria might be seen as an immutable condition, risking prejudice and the perception of individuals with gender dysphoria as abnormal. Additionally, the current classification of gender dysphoria as a psychiatric disorder, rather than a medical condition, can stigmatize those affected, portraying them as ill rather than simply different. This classification would be unjust if gender dysphoria has biological roots, as individuals would be labeled mentally unwell due to factors beyond their control. Furthermore, biological explanations could be misused to justify discriminatory practices or policies, such as denying gender-affirming care or reinforcing harmful stereotypes if certain biological characteristics are not found in individuals claiming gender dysphoria. This is a limitation because it suggests that biological explanations for gender dysphoria might oversimplify the condition, potentially leading to stigmatization and misuse. A limitation of biological explanation of gender dysphoria is that they suffer from biological reductionism. These explanations focus on specific biological factors, such as the size of a particular brain structure or hormone levels, as primary determinants of gender identity. By emphasizing these biological components, reductionist accounts can overlook the complex interplay of psychological, social, and environmental factors that also significantly contribute to the development of gender dysphoria. For instance, while differences in brain structure might correlate with gender dysphoria, they don't account for the diverse personal experiences, cultural influences, and individual psychological processes that shape gender identity. This is a limitation because it suggests that biological explanations alone are insufficient for fully understanding gender dysphoria. A limitation of biological explanations of gender dysphoria is that they suffer from biological determinism. These explanations often imply that gender identity is fixed and irreversible due to biological factors. This deterministic view is problematic because it doesn't align with the reality that many children who initially express cross-gender identification do not continue to experience gender dysphoria as they grow older. This demonstrates that gender identity development isn't solely determined by biology, and that social, psychological, and environmental factors play a significant role in shaping a person's gender identity over time. This is a limitation because it suggests that the deterministic nature of biological explanations oversimplifies the complex and evolving nature of gender identity, making them an incomplete framework for understanding gender dysphoria. A limitation of biological explanations for gender dysphoria is their lack of scientific rigor. It's difficult to establish a clear cause-and-effect relationship between biological factors and gender dysphoria. For example, while studies may identify differences in brain structure or hormone levels between individuals with and without gender dysphoria, it's challenging to determine whether these differences are the cause or the result of gender dysphoria. It could be that these biological variations pre-existed and contributed to the development of gender dysphoria, or they could be a consequence of living with gender dysphoria and its associated stress and experiences. This is a limitation because it suggests that the lack of a definitive cause-and-effect relationship and longitudinal studies undermines the scientific validity of biological explanations. A limitation of the hormonal explanation for gender dysphoria is the existence of research that contradicts the explanation. Some studies have shown minimal hormonal differences between individuals with and without gender dysphoria. Gladue, 1985, found limited evidence of significant hormonal differences between individuals with gender dysphoria and those without. This challenges the assumption that hormonal imbalances or variations are a primary cause of gender dysphoria. If hormonal factors were a direct and significant cause, we would expect to see more consistent and pronounced hormonal differences across individuals with gender dysphoria. This is a limitation because it suggests that hormonal factors play a less central role than some theories propose, and their influence is more complex and intertwined with other biological, psychological, and social factors. Social explanations of gender dysphoria. Social explanations of gender dysphoria take a learning theory approach, behaviorist and social learning theory, and suggest that gender dysphoria is learnt from the environment through socialization. The social learning theory suggests that children learn gender dysphoric behaviors through identification, imitation, and modeling, and these behaviors are maintained through operant conditioning via positive and negative reinforcement. Many young children will observe and imitate their opposite-sex parental role models' behavior, and through this will experiment with gender role behavior. For example, a young boy might observe his mother receiving compliments on her dress, vicarious reinforcement, and imitate this behavior by wearing a dress or jewelry. A young girl might observe her dad being desired by her mother for being dominant and working, vicarious reinforcement, and imitate this by pretending to be dad when playing games that imitate families. Parents or other adults may encourage this behavior through reinforcement, and children may be rewarded for displaying and exhibiting cross-gender behavior. For example, a boy may be complimented and praised for wearing a dress, or a girl may be respected for showing confidence and leadership skills in a family situation. This creates a conflict between the child's anatomical and biological sex and psychological gender, leading to gender dysphoria. Conditioning experiences may explain why more children than adults are identified with gender dysphoria. It may be that because early life experiences are dominated by the family, where cross-gender behaviors are tolerated or even encouraged, influence the diagnosis when young. It may therefore also be that as they grow up and people outside the family exert an influence, prejudice, bullying, or even stigmatization that prevents them gaining employment, prevents people from expressing such a disorder as they opt to hide it. A strength of social explanations of gender dysphoria is that they are supported by research. Riekers and Lovars, 1974, evaluated the behavioral treatment of deviant sex role behaviors in a male child. They investigated the use of reinforcement and punishment to target non-gender conforming behaviors of a 5-year-old male child. The researchers coached the parents to praise the boy when he played with masculine-associated toys and ignore the boy and give him a red token when he played with feminine-associated toys. These red tokens could result in any of 3 outcomes: losing a blue token, going into timeout, or being spanked by his father. The researchers would reassure the parents, e.g., "You're doing the right thing." They observed the behavior changes that they sought, but that the boy's masculine behaviors were not generalizing to situations when he was alone. They also found that the most effective form of punishment for the boy's behavior was when the red tokens were backed up by spanking. This is a strength because it suggests that gender-related behaviors can be shaped through external influences such as reinforcement and punishment, highlighting the role of social factors in the development of gender expression. A limitation of Riekers and Lovars, 1974, study is that it breaks ethical guidelines. Riekers and Lovars, 1974, directly facilitated the shaming and abuse of a 4-year-old child because he engaged in behavior that was not judged to be appropriate for his sex assigned at birth. The authors further used disturbing sexualized language, i.e., fantasies, flirtations, slovenly, seductive eyes, when describing this preschool-age child in the corresponding published article. This is a limitation because it suggests that the study's findings may be compromised due to the ethical violations, raising questions about the validity and generalizability of the results. A limitation of Riekers and Lovars, 1974, study is that it was hugely criticized by other researchers around the time of publication. Nordyke et al., 1977, began their written response by asserting that Riekers and Lovars, 1974, paper is not only accepting but also supporting sex role stereotyping, thereby failing to contribute to the solution of a larger social problem. This is a limitation because it suggests that study was observably unethical and unjustifiable when it was being carried out, and despite this, the study still went ahead and was published, highlighting the rigid ways in which transsexualism was viewed. A limitation of Riekers and Lovars, 1974, study is that it is socially sensitive. The study suggests that the best way to ensure that a child does not develop gender dysphoria is to abuse them. The study has subsequently been used as empirical support for conversion therapy. This is a limitation because it suggests that the study's long-term effects were detrimental, causing significant psychological harm to the participant. A strength of social explanations of gender dysphoria is that there is longitudinal research that suggests individuals grow out of the disorder after being influenced by social factors. Zucker et al., 2008, conducted a longitudinal study on gender dysphoric females and found that of those referred to a clinic between the ages of 2 and 3 years old, only 12% of females were still gender dysphoric as adults. Only 20% of males were still gender dysphoric. This is a strength because it suggests that social factors play a significant role in the development and resolution of gender dysphoria. A strength of social explanations of gender dysphoria is that research for gender dysphoria from twin studies suggests that there must be social factors that affect gender dysphoria. The fact that identical twins, who share 100% of their genes, do not always share the same gender identity or experience gender dysphoria, shown by concordance rates for gender dysphoria amongst identical twins being lower than 100%, Halens et al., 2012, 39%, indicates that environmental or social influences must be contributing to the expression of gender dysphoria. This is a strength because it suggests that social factors play a role in the development of gender dysphoria, as evidenced by the incomplete concordance rates observed in twin studies. A limitation of social explanations of gender dysphoria is that real-world knowledge contradicts the explanation. Transgender individuals often face many social difficulties, e.g. prejudice and bullying, and if social explanations of gender dysphoria are correct, this should discourage cross-gender behavior. However, the fact that many transgender people insist on living as their preferred gender despite these negative consequences. This is a limitation because it suggests that social explanations of gender dysphoria may not fully account for the persistence of cross-gender behavior among transgender individuals who face significant social challenges and discrimination. A limitation of social explanations of gender dysphoria is that they are environmentally deterministic. Environmental determinism implies that social forces are the primary or sole determinants of gender dysphoria, which may overlook the complex interplay between environmental, biological, and psychological influences. This narrow focus on environmental factors may lead to an incomplete or reductionist understanding of the multifaceted nature of gender dysphoria, failing to account for the unique experiences and individual differences that contribute to its development. This is a limitation because it suggests that social explanations of gender dysphoria may overemphasize the role of environmental factors in shaping gender identity development, potentially neglecting the influence of biological or individual factors. Psychosocial explanations of gender dysphoria. There are two social psychological explanations for gender dysphoria: psychoanalytic theory, psychodynamic, and dual pathway theory, cognitive. Psychoanalytic theory of gender dysphoria was proposed by Ovsey and Persson, 1973, and later supported by Stoller, 1973. This theory suggests that gender dysphoria amongst males is caused by extreme separation anxiety from the mother figure before gender identity has developed, e.g. during the critical period, first 2 years of life. To reduce and relieve this anxiety, the boy fantasizes a symbiotic fusion with his mother, by imagining and becoming the mother. This leads to greater female identification and confused gender identity. A strength of the psychoanalytic theory of gender dysphoria is that it is supported by research that found a link between gender dysphoria and an overclose relationship mother and son relationship, which implies a child may be experiencing extreme separation anxiety. Stoller, 1975, observed clinical interviews with individuals diagnosed with gender dysphoria and found that many individuals displayed overly close mother-son relationships. He concluded that gender dysphoria results from distorted parental attitudes, with the potential to lead to greater female identification and confused gender identity. This is a strength because it suggests that there is empirical evidence supporting the psychoanalytic theory of gender dysphoria, indicating a potential link between early childhood experiences and the development of gender dysphoria. A strength of the psychoanalytic explanation of gender dysphoria is that there is research to support idea that cross-gender fantasies are used as a means of resolving the anxiety. Coates et al., 1991, proposed that gender identity disorder is related to mental illness, which in itself is linked to some childhood trauma or maladaptive upbringing. They presented a case study of a boy who developed gender dysphoria after his mother had suffered from depression following a miscarriage when he was 3 years old. They suggested that the boy had developed gender dysphoria as a defensive mechanism to deal with the anxiety experienced by the trauma. This is a strength because it suggests that psychoanalytic explanations offer a framework for understanding how early childhood experiences and psychological trauma can contribute to the development of gender dysphoria as a defensive mechanism. A strength of the psychoanalytic theory of gender dysphoria is that it is supported by research that found a link between gender dysphoria and disordered attachment, which concerns the same emotional bonds and distress experienced in a relationship as separation anxiety. Zucker et al., 1996, conducted a study using 115 boys that had concerns about their gender identity were studied along with their relationship with their mother. 64% were diagnosed with separation anxiety disorder along with gender dysphoria compared to 38% whose symptoms were subclinical. This is a strength because it suggests that the emotional bonds and distress experienced in a relationship, as reflected in disordered attachment and separation anxiety, may play a significant role in the development of gender dysphoria. A limitation of the psychoanalytic theory of gender dysphoria is that it is considered limited and gender bias, as it does not explain atypical development in females. The theory only offers an explanation for the development of gender dysphoria in males, because females, according to this theory, do not feel the same pressure to take on the role of the mother ir fear of separation. This idea was supported by Riekers, 1986, who suggested that gender dysphoria in males is more motivated by the lack of a father during the critical period for attachment formation, as opposed to an absent mother. This is a limitation because it suggests that the psychoanalytic theory cannot be generalized to females as it overlooks important factors and experiences that are specific to females. However, Zucker, 2004, did attempt to create an explanation for females through the father-daughter relationship and suggested that females identify as males because of severe paternal rejection from their fathers in early childhood, and unconsciously they think that if they become males, they might gain acceptance from their father. A limitation of the psychoanalytic theory of gender dysphoria is that it is unfalsifiable. The psychoanalytic theory is based on Freud's theories of unconscious concepts and conflicts that are the driving force behind gender development. These concepts cannot be proven wrong, as these constructs are not directly observable or measurable, which poses challenges for empirical investigation. This is a limitation because it suggests that the psychoanalytic theory of gender dysphoria lacks empirical testability, making it difficult to validate or refute its claims through scientific methods. The dual pathway theory of gender dysphoria was proposed by Lieben and Bigler, 2002. They suggested that children develop gender schemas through two pathways. 1. The gender schema pathway, where children learn about gender through societal norms and expectations, forming rigid gender schemas that influence their behavior and attitudes. 2. The interest-based pathway, where children develop interests and preferences that are not necessarily tied to gender stereotypes through activities and their own interests, allowing them to form non-sex-typed schemas. If this second pathway is dominant, this may cause the schema to be affected, leading to androgyny or gender dysphoria in a minority of children. A limitation of the dual pathway theory is that it is a limited explanation. While the theory proposes the existence of two distinct pathways leading to gender dysphoria, it does not adequately explain why one pathway may become more prominent or influential than the other in certain children. This lack of clarity regarding the determinants of pathway dominance limits the theory's ability to provide a comprehensive understanding of the diverse developmental trajectories associated with gender dysphoria. This is a limitation because it suggests that the dual pathway theory of gender dysphoria may not fully elucidate the mechanisms or factors that influence the relative dominance of the two proposed pathways in different individuals. Thank you for watching. If you have enjoyed this video, please like and subscribe for more content like this. Bye.